Greetings and welcome to the Tetra Bio-Pharma Incorporated conference. It is now my pleasure to introduce your host, Chief Executive Officer Guy Chamberlain. Thank you, Mr. Chamberlain. You may begin.
Thank you very much. Welcome, everyone. Also the Chief Regulatory Officer of Tetra Bio-Pharma. We are a publicly traded company, so you can have a look at our forward-looking statement, but basically it is a pretty standard statement that most public companies will provide shareholders and investors. As the company, obviously we have a very strong team in drug development that has many years of experience in regulatory affairs, developing the actual protocols, executing them, overseeing them with clinical research organizations, and this includes on the compliance front. That's a very key aspect to ensure that all the clinical sites run adequately from a regulatory point of view. We have also a global intellectual property portfolio with patents that extend out to 2034. This has been part of our model to be sure that as a biotech company, all of our drugs were protected with patents. Many of these patents are granted. They are not pending. Our ARDS-003 drug, which we hope will enter phase 2 clinical trials in Q4 of 2020 for treating the cytokine release syndrome and acute respiratory distress, is our lead and will be the main focus of my talk today. Like I said, it is our lead asset. We hope to make it available by mid-next year in the global fight against COVID-19. We also have some other drugs. Our Quixley is in a trial that's been approved by the FDA, phase 2 trial, and this obviously will be proceeding in the coming months. And as we announced this morning as well, we're looking to launch a phase 3 trial of this the Quixley product in Europe. As a company, our capital structure, well, we trade our ticker symbol is TBP, and in the U.S. on the OTCQB is TBPMF. We have a market capitalization, obviously that varies day to day depending on our share price, but it can be these days anywhere between 60 and 80 million. We have a cash position of about a bit more than 11 million. We have an experienced management team. There's myself. I've been developing new drugs since 1995, so about 25 years of experience. We have a CFO, Mr. Diapalo, who has been around, I guess, in this industry, financing biotech companies and other smaller companies for many years. Steve Neron, who has joined the company to be responsible for commercial operations, comes from Pfizer, Merck, and Bush Health, and has over 35 years in commercial operations. And our Chief Scientific Officer is Dr. Melanie Kelly. She's also professor at Dalhousie University and has been studying the cannabinoid system for a few years, more like 20 years. Our drug development pipeline, which is available also on our website, I'll just point out several of them. Like I was saying before, our Quixley is in entering its phase 2 trial, and in parallel we'll be starting our phase 3 trial in Europe. And we have other drugs going. One of the key ones we'll potentially hear more about later this autumn will be the Reborn trial, which is a drug that's designed basically to go after breakthrough pain. So it's really speed of relief. It's really designed for that, contrary to many other medications, oral, even, like I say, fast release formulations will not provide as fast of a relief as an inhaled drug to basically prevent the pain. So this will be actually a very important study, especially if you're looking at the fight basically against the use of opioids. Our ophthalmology program will also be beginning in 2021, right after we complete the phase 1 study of our COVID-19 drug. So it is actually the same active drug substance. So these two actually will move in parallel. And then we have our hepatocellular carcinoma drug that will eventually move. We obviously move products based on priorities. We typically move one fast priority and then followed by a second one, and then all the other indications move at slower speeds based on our ability to finance these operations. So our new priority, which is the ARDS-003, a COVID drug, it's actually a unique drug. Our team has been studying sepsis and actually trying to prevent sepsis or treat sepsis for over 12 years. This includes research on, you'll see some of this data I'll present it, but this is a key aspect, contrary to many other companies that are repurposing anti-inflammatory drugs for trying to prevent the cytokine storm. Our team has actually been studying this cytokine storm and the damages that it causes, like causing organ dysfunction, even leading to pulmonary fibrosis and acute respiratory distress syndrome for over 12 years. So we believe our ARDS molecule is a well-designed molecule because it was specifically developed for this indication, and we're not repurposing it for COVID-19. So this really makes us as a unique company. Everything we do from a manufacturing and clinical point of view is all done here in North America. We do not have any outsourcing into any other country for these drugs. So the immune response, just for those of you that are not really familiar with it, and basically the CB2 receptor, but basically if there's an injury to a tissue or an infection, basically it will trigger an inflammatory process. And what happens basically is the immune cells will activate systems within the body to basically cause the inflammation and come in and basically try and fight off the disease or the damage. And you'll see some of these cytokines, interleukin-8, TNF-alpha, these are all cytokines that are responsible, either pro-inflammatory molecules that are basically involved in this inflammatory process. So this is what this slide is showing. So there are two phases to the immune dysregulation in sepsis. The first one is really the key one to understand. You can look at it as kind of like a systemic inflammation that's really an overactive immune system that leads to the damage. Simply put, basically once you're in this hyperinflammatory phase, the inflammation will decrease the blood flow to some of the organs, and this will cause damage to these organs. And obviously now that you're in this hyper phase, it is followed then by an immune suppression, which is you've kind of now exhausted your immune system, and this is where the deaths come in, and you get the hospital-acquired infections or the resurgence of an infection because now your immune system can't fight it. And this is what we're facing in COVID. So basically just to kind of show you graphically what it is. So you have this hyperinflammatory, which is a cytokine release syndrome. It's involved in many other conditions, not just, say, a viral infection or bacterial infection. And then it's followed by this down phase. And this is where once the immune system is suppressed, that you will actually get the deaths that occur in the patient. So this is why it's key to be able to have a drug that acts on this hyperinflammatory phase, because this will help, obviously, clinicians in managing during a pandemic the COVID cases. Because, for example, if you're getting multiple patients coming in that are severely infected, you don't want them all to go to ICU. So if you can give a drug and rapidly decrease this hyperinflammatory response, then you can help decrease the number of patients that will actually require the ICU or the ventilator. And the same thing if you treat people even early on, you can actually prevent the hospitalization. So this is what our goal is to demonstrate in the clinic. So our sepsis treatment is basically there's none that have been approved yet by the FDA or Health Canada. Basically it will involve giving it with antimicrobials with a standard of care, which is intravenous fluid, and helping manage whatever organ dysfunction a patient may see. When COVID started bringing patients to the hospital, people were only seeing the ARDS, the acute respiratory distress, but as patients, like I say, began coming out of the ventilators, we were seeing basically some liver injury, kidney injury, intestinal injury, even heart injury. So this is all part of this cytokine hyperinflammatory response that occurs in these patients. This is why a drug like ours is key to get to the clinic as fast as we can. So obviously, most of you have heard, obviously, the coronavirus disease, which is caused by the SARS-CoV-2 virus. I won't mention much more about this because obviously it's well known. We always think, like I said, just about the pneumonia, that it basically progresses to the acute respiratory distress syndrome, but also keep in mind that there is a multi-organ dysfunction syndrome that can also lead to death and does occur in these patients. Our models, which you'll see very shortly, they're based on endotoxin and bacterial sepsis. So we have over 11 peer-reviewed publications in scientific journals that were done by the team over the last 12 years. They weren't done, say, with viruses, but basically the hyperinflammatory phase of sepsis is the same, be it a virus or be it a bacteria or be it a chemical injury. The body doesn't have a specific system for each of these. It is the same one. So this is kind of to show you the validity of our data. So like we're showing here, is this disorganized systemic immune response, that's this acute response, and then followed by obviously our sepsis. So the endocannabinoid system, most people are not familiar with it. In short, basically, there's two types of receptor, or CB1 receptor, or cannabinoid 1, and then there's the cannabinoid 2 receptor. But cannabinoid 2 is the one we're really interested in because this is the one that's basically expressed on the immune cells in the body. So when we target the CB2 receptor, we're actually targeting the immune system in the body. So that's a key aspect to understand. So the endocannabinoid system is a target for treating inflammation and fibrosis. Obviously, if I have an agonist, so I activate the type 2 receptor, which is on the immune cells, I can basically start to manage the hyperinflammatory stage of the experimental sepsis. So this is why we will use agonists against the CB2 receptor. Now, if I want to do something different to it, kind of like block my response, which we'll see, then I'll use an antagonist of this receptor. In the case of the CB1 receptor, it is only an antagonist of the CB1 receptor that can have use in, for example, decreasing the inflammation and decreasing the potential fibrosis in chronic inflammatory diseases. So our ARDS indication and treatment, so our drug is basically called HU308. It's a drug that's being developed for both cytokine release syndrome, so the hyperinflammatory phases of sepsis, and for obviously COVID-19, because obviously beyond COVID there are other indications, so we will be going after these other indications as well. The drug is currently developed as an intravenous bolus injection, and this is because we're targeting obviously inpatient care for this condition. So our treatment phase, so here's kind of a symbolic representation of what will happen with our drug. So our drug will reduce this hyperinflammatory phase, and now because obviously I've been able to decrease the exhaustion of the immune response, I will now get a less pronounced immune suppression, and therefore our goal is to prevent the immune suppression, prevent the acute respiratory distress, and even prevent the organ damage in patients. So this is the concept of how our drug will work. So our team went out and showed it. This slide is also part of a publication, and it basically, they use a model of animals where they kind of genetically deleted the CB2 receptor from these cells, and they were able to basically to validate and show the role of the CB2 receptor in the cytokine release, which was a key part to validating all their work that they were to do the next, say, 11 years later. So here in this slide, we're basically looking at what we call the adherence of basically leukocytes in the intestinal submucosal venules. And what you're seeing here by the slide is our drug referred to here as the HU-08. The LPS is what induces the basically the blue you're seeing is a significant reduction in the leukocyte adhesion. It's about by 57%, and with a p-value of 0.0001, which is pretty significant. And then they use an antagonist to kind of show that if you then block the action of the drug, that you actually get the opposite effect, which is key in pharmacological research. Our next demonstration is basically showing using markers of inflammatory cell recruitment. These are intercellular adhesion molecule, the ICAM, and the vascular cell adhesion molecule, which is the VCAM. And here basically the goal is a bit the same. It's just showing that our drug treatment will significantly reduce these pro-inflammatory cells. So basically they're coming in and calling in the inflammatory response. And the same thing here in this slide, we're showing the exact same thing, that we significantly reduce these inflammatory cytokine levels. So we're seeing a significant reduction in interleukin-1B, in the TNF-alpha, interferon gamma, interleukin-6. So these are all being significantly reduced by our drug. I don't have it in this slide, but we also compared our drug to dexamethasone in similar models of sepsis, and our drug was shown to be statistically superior in these animal models to dexamethasone. And I'm pointing this out because dexamethasone did have a first proof of concept study that was that did offer some potential benefits to patients that were severely affected by COVID. And the last slide of results, basically to show this to you. So basically here we're showing again the same thing. So if you were looking at a measure of inflammation, which here is basically adherence of leukocytes, you're seeing basically the effect of the drug, a significant reduction compared to the induced sepsis, which is a complete black one, the written LPS. And then antagonizing the effect with another drug, then you basically eliminate the benefit of the drug. So this is a very solid pharmacological evidence of the potential efficacy of our drug ARDS-003. So our new drug will be entering phase 2 late in Q4 2020. We're currently finishing the toxicology studies, the IND-enabling studies. We've had our pre-IND meeting with the US FDA. They validated our program. They did provide very good guidance on our study designs for the clinical trial, and we'll be obviously implementing those designs before filing for the IND. Our model is typically pursuing basically a partnership with any pharmaceutical company, because we don't have a business of sales and distribution of commercial drugs. And upcoming milestones, like I said, we're basically finalizing our toxicology. And then basically be filing then our CTA for starting the phase 1 in humans in Q4 of 2020, which should then be followed if these trials are successful to initiate a phase 2 later by the end of this year. And our other milestones, obviously our Plenitude trial is supposed to the treatment phase in patients, so it's been approved by the FDA. And the VA have licensed all of our sites, so this study should be moving as per our other communiques to the market. And in 2021, well, this is where we're hoping by mid-2021 we'll be in a position to have demonstrated the potential of our ARDS drugs in patients and make it available for emergency use to physicians. And on the other fronts, our other drugs will obviously be moving, so our Serenity trial should begin to move. Our Reborn trial should have been started also by then on the clinical front, and we should be finalizing our Plenitude trials. And notice that there are two. There is a phase 2 Plenitude trial, which is run in the United States, and now, as we announced this morning, we'll be applying for a phase 3 trial basically in Europe to run the next study of quick-sleep, and this is for pain indications. And in 2020, our goal is to expand from ARDS to other cytokine release syndrome indications. At this point, I will take some questions. Well, the question that's being asked, there are approximately 165 vaccinations being developed for COVID. Do you worry about that a viable vaccination would displace the relevance of ARDS 003 research? This is actually a very, very good question. We do get that one often. Unfortunately, if we look at the success of influenza vaccines over the years, the most efficient, let's say, batches of a vaccine for influenza have been just about efficacy of about 60%. They rarely go, say, to 90%. There have been, I believe, measles or other diseases that have had the success of over 95% efficacy in the patient population. So, I mean, from a humanitarian point of view, I do hope the success of one of these 165 to be able to bring us success of at least 95% of the population to be protected, and also that it covers a region of the virus that will not have any mutation. Unfortunately, like we've seen in the influenza vaccine research for years, decades, is that this does not occur. They have never been able to develop a universal vaccine for influenza, and this is why we always have to vaccinate every year. And we do see cases of ARDS from influenza. So, I mean, the ARDS indication, for example, is present in not just COVID, it's present with influenza, it's present in sepsis. So this is kind of our goal. We know it won't disappear, and there's other indications that we have not revealed that we will be targeting because there are many other cytokine release syndrome diseases. The next question is, what is the difference between the United States Phase 2 and the European Phase 3? Basically, the difference will be in the number of patients and how it is powered. But it is the same indication that we are going after because we would want to add to our claims for pain management. The question is, any infos on the possible on our Canadian grant application? To date, we are still, I guess, a potential candidate. We have not have answered all the stages of questioning, and at this stage, we're just waiting for the response. The next question, what are you doing in the short term to prop up the share price? As you know, it has been sliding towards the downside over the past 8 to 9 months. That's also another interesting question. One of our goals had been to do an upgrading to the main TSX, which we just achieved on August 4th, so we are now no longer listed on the venture. There is a lot of activities that kept our share price down that should be eliminated on the TSX, plus the fact that it is a graduation. It invites institutional investors to Tetra, which should help stabilize the price. One of the problems we have as a company is obviously that our share price is held by mainly 90-something% retail and not institutional. By switching and upgrading to the TSX, we can then get institutional investors. It also brings on more long-term investors to the company that will help stabilize the price. And obviously, as a company, which I can't comment what's coming in the news, because that would be a selective disclosure, but basically, as a company, we obviously you've seen our milestones, so we hope to be achieving them on time and bringing very positive news that will translate into increase in share price. And I want to thank everybody for attending my presentation, and I hope that you enjoyed it and learned a bit more about our ARDS drug development program. Thank you very much.
This does conclude today's conference. You may disconnect your lines at this time. Thank you for your participation.
Thank you.